Ready.
A very good morning to all of you. Now, last class we have discussed how we will dissect out the frog and how does a normal cardiogram look like. So today we will go to the experiments. Okay. Now before we go into the experiments, you have to know the principle. So this experiment will work on the principle that if the beating heart of a frog is connected to a liver, then its contraction and relaxation can be recorded on a smoke drum. And the effect of temperature, drugs and ions can be studied. So this experiment has three parts. First we will study the effect of temperature, then we will study the effect of ions, next we will study the effect of drugs on the frog's heart. So first we will talk about the effect of temperature on frog's heart. Now before going into the discussion proper, one thing you have to know, what will represent the force of contraction in the cardiogram and what will represent the rate of contraction. Now if you remember, like if I am showing it with the isotonic lever, okay, if I am showing it with the isotonic lever, the normal cardiogram has two peaks. One is of the sinus venosus and the auricle, and other is of the ventricular systole. Okay, so you remember in the isotonic lever, the upstroke is representing the systole and the downstroke is the diastole. So the normal cardiogram, we get a curve in this fashion. Okay. Now if it is with the Frank-Starling's lever, then you know the downstroke is a systole, the upstroke is a diastole. So again you get a curve like this. Yes. Okay. So these are the normal cardiograms. Now you see what will represent the force of contraction. Now the height of the curve represents the force of contraction, and the number of waves that we get, okay, in 1 second or in 1 minute, that will represent the rate of contraction. Now to see the rate of contraction, you have to take a time tracing so that you can count the number of waves that comes in 1 second or number of waves that is coming in 1 minute. Okay, so that is how you count the rate of contraction, and the height will represent the force of contraction. Now coming to the experiment, that is the effect of temperature. Okay, so here the dissecting out the frog is same like the dissection. Now after you dissect out the frog, what we do is we take Ringer's solution. Now Ringer's solution is isotonic and isoosmotic to the frog's heart. So we will take Ringer's solution. We take two types of Ringer's solution. One is the hot Ringer's at a temperature of around 34 degree to 36 degrees Celsius, and other we will take as the cold Ringer's at a temperature of 0 to 5 degrees Celsius. Okay. Now here what will you do? What we will do is that first we will take a normal recording. I am showing it with your isotonic lever because the curves that are present in our lab are with the isotonic lever. Okay, so we'll take a normal cardiogram. Okay, we'll take a normal cardiogram recording. Okay, we'll take suppose 2 to 3 beats. Okay, first we'll take a normal cardiogram recording. So this is with the isotonic lever. Now with the Frank-Starling's lever, the same recording will be like this. Okay. Now what we will do, we will pour warm Ringer's solution over the frog's heart. Warm means at a temperature of 34 to 36 degrees Celsius. Now once we put the warm ringer solution, we wait for few minutes and we put a temperature and see whether the frog's heart has come to that temperature. Now once it comes back to the temperature of the ringer solution, now we again record a recording of the cardiogram. So again we will take a recording. Okay, we will take a recording. Similarly, this side also we will take a recording. Okay. Now after we take a recording, next we wash the frog's heart with again your normal ringer solution at a normal room temperature, and we keep on washing till again the heart comes back to the normal room temperature. Okay, so once it comes back to normal room temperature, again we take a recording. Okay, we take a recording. Similarly, here also we take a recording. Okay. Next, after it comes back to room temperature, now we will give cold ringers at a temperature of 0 to 5 degrees Celsius, and we wait for again few minutes to check whether the heart is coming back to the temperature of the cold ringers. Now once it comes back to the temperature of the cold ringers, next we take a cardiogram reading. Here again we take the reading. Similar for this also we take a cardiogram. So this was when we have given your hot ringers. This was when we have given our cold ringers. Yes, this is H for hot, C I have written for cold, and these two are the normal. Okay, these two are the normal recording. Now you see what is the effect of temperature. Okay, now what is the effect of temperature? Now we have seen that as we have given hot, okay, as we have given hot ringer, that means we have increased the temperature, we have found, you see, the height of contraction is increasing. That means there is an increase in the force of contraction. Yes, and also you see the number of waves that are coming at a faster rate. Therefore, that means the heart rate or the rate of the heartbeat is also increasing. So we see with the effect, with the increase in the temperature, we find both the force of contraction as well as the rate of contraction is increasing. Okay, but here you see, you come to the cold ringers, you see the height is decreasing. That means the force of contraction is decreasing, as well as you see the spacing has increased. That means the rate of the heart rate has also decreased. So increase in temperature causes an increase in the force of contraction as well as increase in the rate of heart rate, as well as the decrease in the temperature causes a decrease in the force of contraction and decrease in the heart rate. Now why are we getting this? Now as soon as you increase the temperature, what happens? Temperature increases the permeability of the nodal tissue to sodium. So what happens? Sodium will increase inside them. So if sodium increases, what will happen? The heart rate will increase. And why is there increase in the force of contraction? Because with increase in temperature, the viscosity of the cardiac muscle will decrease, and moreover, the enzymatic activity of the muscle proteins will increase, which will lead to an increase in the force of contraction. And the vice versa occurs in cold temperature. As a result, we get a decrease in the force of contraction and decrease in the heart rate with cold temperature. And here one thing has to be noted, that you have to give always the hot ringers before giving the cold ringers. Now that is because if you give cold ringers before giving hot ringers, what will happen? This cold ringer decreases the enzymatic activity, and sometimes it decreases and it causes a deactivation of the enzymatic activity, and then after recording the cold ringers will not be equal to record the warm ringers activity. Therefore, you always give hot ringers before giving the cold ringers. And one thing you have to remember here also, that the temperature should not be more than 36 degrees Celsius when you give the hot ringers, because if you give it more than 36 degrees Celsius, what will happen? The cardiac muscle will undergo denaturation and the muscle will go into heat rise. Okay, so that is the effect of your temperature. Now, your cardiogram is this. Okay, so this is the cardiogram. If you see the cardiogram, this is the where it is written N. This is the part where you are getting the recording of the normal cardiogram. The C, if you see, that is the cold ringers. You see the height has decreased, as well as the spacing between the impulses have also decreased. Okay, that means the spacing has increased, as well the height has decreased. That means the heart rate has decreased and the force of contraction has decreased. But where it is written as H, you see the height has increased. That means the force of contraction has increased, and moreover the spacing has decreased. That means the rate of heart rate has also increased. So if this is given, you have to say that this is a cardiogram showing the effect of temperature on the frog's heart. Thank you.